Welcome to this introductory session on proteins. I'm sure all of you recognize at least a few of the food items from the image displayed here. You have fruits such as avocados and pomegranate, pulses such as mung beans, vegetables such as cabbage and spinach, and nuts such as almonds. All of these are known to be rich in proteins. And we are all aware that proteins are required by the body for several functions. So let us now consider some of the important functions that proteins perform in our bodies. Some proteins support the regulation and expression of the nucleic acids DNA and RNA. Some proteins are antibodies that support the immune function and thereby enable your body to fight against invading bacteria and viruses. Some proteins function as digestive enzymes that help facilitate chemical reactions in the body and therefore help you get energy from the food that you eat. Some proteins are found in the muscle, where they support muscle contraction and movement. Some proteins are structural proteins that provide support to the body. And further still, some proteins function as hormones that help coordinate bodily function. And lastly, you have some proteins that move essential molecules around the body. A classic example of this is hemoglobin, which helps transport oxygen from one part of the body to another. Having understood the significance of proteins in your body, I'm quite sure that you're curious to know that at the molecular level, what exactly are proteins? Proteins are polypeptide chains made up of building blocks of amino acids which are linked to each other by a peptide bond. In the diagram that is shown here, the beads or the spheres represent individual amino acids, and these are now linked via the string, which is nothing but the peptide bond. Let us therefore now look at what are amino acids. Amino acids are molecules that contain two functional groups, namely an amino group and a carboxylic group. Every amino acid has a basic structure as shown in the image on the left section of the slide. Every amino acid has an alpha carbon to which four distinct groups are attached: a carboxylic group COOH, an amino group NH2, a H group, and a specific R group. It is a variability in this R group that gives a variety of amino acids. Based on the nature of R groups present, amino acid can be classified into 5 different groups. The first group considered here is the non-polar aliphatic amino acids that include glycine, alanine, valine, leucine, methionine, and isoleucine. The next group is that of polar uncharged R groups. This includes serine, threonine, cysteine, proline, asparagine, and glutamine. The third group of amino acids are those that have positively charged R groups, which include lysine, arginine, and histidine. The fourth group of amino acids are those that contain negatively charged R groups, which include aspartate and glutamate. And finally, the last group of amino acids, which contain non-polar aromatic R groups, which include phenylalanine, tyrosine, and tryptophan. Having understood the structure of amino acids, let us now look at the different levels of protein structural organization. Although in the beginning of the session we mentioned that proteins are polypeptide chains made up of amino acids linked via peptide bonds, in reality, the functionality of the protein goes much more beyond this simple definition. A protein is said to be functional or having its native structure only if it has folded to an appropriate conformation that relates to a function. Thus, the levels of protein folding are very important to protein function. The first level, referred to as a primary structure, is the one which basically refers to the sequence of amino acids from N terminus to C terminus linked via peptide bonds. This is a result of mRNA translation carried out by ribosomes. This primary structure then undergoes local folding to produce the next level of organization known as a secondary structure. There are two types of secondary structures, namely the alpha helix and the beta sheets. The alpha helix here has been represented as coiled ribbon, whereas the beta sheet has been represented as ribbons with arrows which have been placed one below the other. Several of these secondary structures then interact with each other across distance to form the tertiary structure. For a protein with a single polypeptide, this tertiary structure is in fact the highest level of organization. However, for a protein with more than one polypeptide, each polypeptide that had folded to their respective tertiary structures will then interact to form the quaternary structure. Having understood the basics of protein, I now invite you to explore an educational portal known as the PDB 101. To do so, just type PDB 101 in any of your internet browsers. The very first page that is shown out here is a PDB 101 home page, and if you were to click on it, it takes you to the home page. And what is it that you see? You see a page filled with resources for you. Now, these resources are learning resources, equally important for learners as well as for tutors. Now, if you want to search anything on this site, you just have to type whatever you want to look for in this particular section. Or, an easier way to do this is to browse by different categories. For example, you have 4 major categories of health and disease, molecules of life, biotech and nanotech, and structures and structure determination. Another important feature of this PDB 101 is known as the molecule of the month. If you were to click on this particular tab, you will be taken to the highlighted molecule of the month. And for April 2020, that molecule is a photosynthetic supercomplex or light harvesting complexes. You have a lot of information which has been given about this complex, and additionally, you also have the image in the form of a 3D structure which you're now more comfortable with. If I were to click on this JSMOL tab, you will now be directed to an image which is a little bit more interactive. Once it's loaded, you will understand what I'm saying. Well, then you now have the structure that is loaded. You're probably going to say that the structure looks exactly like the one that was shown in image. So here comes the magic. Take your mouse and place it on the structure, left click, and then just start moving. And what do you see? You're able to actually rotate the molecule, and you're able to see this particular protein from all different dimensions and angles. Now isn't this just awesome? Apart from this particular resource, there are several other resources which have been put on this site. As a learner, what should probably interest you are all these resources which have been put under the Learn tab. You have resources such as paper models, flyers, posters and other resources, videos, interactive animations, coloring books, and many more. Let us now look at one or two of these in detail. If you click on the paper models, you will be directed to a page which shows you a number of paper models. Let us now look at one of these. If you click on the green fluorescent protein, we'll be directed to a page which will show you how to make a paper model of this particular protein. Now, what is a GFP? The GFP, or the green fluorescent protein, is a protein which is found in jellyfish, and in fact, it is a bioluminescent protein. So are you all set to make this particular protein? To do so, there are several instructions which have been given. If I were to click on this download and print template PDF, you will be directed to a particular PDF which gives you instructions. Page 1 gives you stepwise instruction how to go from the template to the final folded structure of the protein. Page 2 provides the actual template which you will be printing, and you will then start cutting. Now, just in case you have not understood any of this, or probably at some particular step in between you're getting stuck, you're not a self-assured, therefore you do not know how to fold a protein, don't worry, PDB 101 has an additional aid for you. That is in the form of a video, which will now assist you in folding your protein from step 1 to the final step where voila, you have a GFP with you. Now, another resource that I would like you to explore apart from paper models is that of flyers. And one such important flyer which should be of interest to you as a beginner is that which tells you about what is a protein. If you were to click on this and then click the download PDF, you will be directed to the PDF of this particular flyer. The page 1 represents the information of what is a protein. It also gives you information about the primary structure, the secondary, tertiary and quaternary structure. Additionally, page 2 gives you information about the shape and, more importantly, the different functions of proteins. Having discussed the features of PDB 101, I now invite you to explore this portal by yourself till our next session. Goodbye and have a nice day till then.